everyone. My name is Yuan Liu, Associate Professor from University of Pittsburgh, co-founder and the CTO of Coloma Therapeutics. We named our company Coloma after a small town in California where the gold rush started. Today, I'd like to walk you through the gold mine we have discovered. For the last 7 years working at the Aging Institute, Professors Torren Finkel, Bill Chen, and I have collaborated with the biotech and pharmaceutical companies for drug discovery. We have developed 2 IND products currently out-licensed for commercial development. We also developed a high-throughput screening platform named Prospector. The Prospector platform leverages the biophysical principle of cellular thermal shifting. Basically, by measuring the soluble protein level change with the temperature gradient, we can generate a melt curve specific to individual protein as shown in blue here. When a small molecule binds to the protein, it can change the protein thermal stability and shift the melt curve as shown in red. We have developed this approach to a high-throughput fashion amenable to drug screening. Since this approach doesn't require the recombinant protein, we can set up the screen assay rapidly, typically within a week, and finish the screening of our in-house library of 300,000 small molecules within 2 weeks, and generate hundreds of small molecule binders to the protein of interest. Then, with diverse functional assays, a subset binders can be further developed into inhibitor or activator, degrader or stabilizer. In the process, while optimizing our platform, when we screened a variety of traditional undruggable protein targets, we established Coloma to further develop the drug candidates we have discovered in our academic labs. Coloma is currently self-financed, has licensed 2 IPs we founders generated through the university, including the small molecule TNFR1 inhibitor that we are trying to raise for the IND-enabling studies in 2026. The reason that we are interested in TNFR1 is because it plays a central role in immune response related to many inflammatory and autoimmune diseases. In the TNF-driven cell signaling, the membrane bound and soluble TNF ligand trimer can bind to the cell surface receptor TNFR1 and lead to pro-inflammatory signaling, which is often overactivated in many autoimmune disease conditions. While the transmembrane TNF ligand trimer primarily activates the cell surface receptor TNFR2 for the protective signaling, especially in immune regulation. By blocking this TNF cell signaling, the current anti-TNF biologics has a $40 billion US market to treat a broad range of autoimmune diseases, including rheumatoid arthritis, Crohn's disease, and psoriasis. With autoimmune continues to be a major health concern, we expect that this market will grow to be $50 billion within 5 years. However, the current TNF therapies have limited efficacy and adverse effects. Because targeting the TNF ligand blocks its interaction with both the TNFR1 and the TNFR2, leading to the suppression of the beneficial TNFR2 signaling. In addition, Sanofi's recent TNF ligand small molecule inhibitor has hit a setback as a monotherapy in the phase 2 clinical trial. Given all the limitations of the TNF ligand-based approach, how about targeting the receptor? Unfortunately, the development of TNFR1 antagonists hasn't been very successful for the last 20 years. The recent TNFR1 antibodies have shown limited efficacy in clinical trials, largely because the antibody can bind to the soluble TNFR1 protein, compete off, and free up more TNF ligand for signaling. So with all these invaluable lessons we have learned, the development of a non-competitive small molecule TNFR1 antagonist has been highly anticipated. But the TNFR1 protein has flat surfaces, lacking well-defined drug binding pockets. It has been very challenging to develop small molecule binders to proteins like this. However, we were able to use the Perspector platform to screen and identify the potential TNFR1 binders. Through multiple functional assays, we have confirmed the 4 classes of hit molecules. In addition, we have developed computational models to rapidly advance our chemistry campaign and improve the compound potency at least 1,000 times within 6 months. Our current advanced lead compound can suppress TNF-induced ICAM protein expression with an IC50 about 20 nanomole. And the compound doesn't work in TNFR1 knockout cells in the presence of TNFR2, which indicates the compound specifically works through TNFR1. Our compound also has great efficacy and admin properties. It can achieve antibody-like efficacy with almost 100% suppression of the TNFR1 signaling, while Sanofi's TNF ligand inhibitor has about 50% of maximal inhibition. The compound also has a clean profile in many off-targeting assays. It doesn't have a drug-drug interaction liability, cytotoxicity, or cardiovascular and genotoxicity. The compound also has excellent oral PK profiles in both rats and dogs. We're currently advancing the chemistry with the goal of nominating the DC candidates in the fourth quarter 2025. Now, we are trying to raise the Series A financing of $6 million for the TNFR1 inhibitor IND enabling studies starting 2026. We plan to file an IND application in the second quarter 2027 and raise the Series B financing for the potential phase 1 clinical trial in the third quarter 2027. We also expect to raise for the platform development with the potential new drug discovery collaborations. I hope you enjoy the tour and find our unique drug discovery approach efficient and scalable. With your support, we can refine the drug gold together to revolutionize the anti-TNF therapy for better efficacy and safety. With that, I thank you all for your attention and happy to take questions.